Good afternoon. One of the many privileges of organizing the seminar series is a privilege to invite friends. So it's a real pleasure to have Maoz Shamir here. Maoz was an undergraduate student here at the Hebrew University studying physics and math. And then he continued here to do his master's and PhD under the supervision of Chaim Sonkolinsky in physics. Then he moved to BU for several years, and since 2008, he's in Ben-Gurion University. Maoz, please.
Stop it, this computer is in you and then
Yeah. So I thought the privilege is access to good coffee, but nevertheless. So thank you for the introduction and the invitation. Does this work? It's a pleasure to be here, and I'm going to talk about a project actually I'm doing in collaboration with Eli Nelkin and with Trevor Shackleton and Alan Palmer. I think it's fair to say that a large part of my scientific education happened here, not in this room, but in this seminar. And as a theoretician, during my first year, I must admit that I understood very little. And as a result, I also remember very, very little. The only thing I do remember is a single talk, and I don't remember the talk. I can't remember the speaker. I don't remember the title. I only remember the joke the speaker told in the beginning of the talk, which makes you wonder, what are you going to remember from my talk in an hour from now? And even this joke was a very corny joke that every student who studied physics as an undergrad knows it. And it's about how biologists do science. It's about a biologist who trains a grasshopper, it's a longhorned grasshopper, to jump on command. So he trains the grasshopper to jump, tells the grasshopper to jump, the grasshopper jumps. He opens his lab book and writes carefully down, day 0, I trained the grasshopper to jump, I told the grasshopper to jump, the grasshopper jumped. Comes back the next day, cuts off a leg of the grasshopper, tells the grasshopper to jump, the grasshopper jumps. He opens his lab book and carefully writes down, day 1, I cut off a leg of the grasshopper, told him to jump, the grasshopper jumped. So it's a lesion experiment, as you all know, and so on and so forth. Second day, cuts another leg. The 6th day, here shows the grasshopper have 6 legs. In the 6th day, he cuts off the 6th leg of the grasshopper, tells him to jump, and the grasshopper does not jump. So he opens his lab book carefully, writes down, day 6, I cut off the 6th leg of the grasshopper. I told him to jump. The grasshopper did not jump. Conclusion, grasshoppers without legs cannot hear. And everybody laughs. And the reason I remember this joke is that then the speaker said something that I actually did not know. He said, the funny thing is that the conclusion is actually correct. So the grasshoppers that, as far as I understand, are termed longhorn grasshoppers, probably chagulim in Hebrew, actually have their say, loosely speaking, hearing organs at the knees, joints of their front legs. Okay? So if you actually cut off the legs, they don't hear. And insects can have hearing organs, and this you can get what the seal was. And another very famous example, which is particularly famous here in Jerusalem, is the escape mechanism of the cockroach. So that also sense a condition of air pressure that propagates to alert him of an attack of some predator. Might it be a pink slipper or a newspaper? Nevertheless, the grasshopper can sense it, and it generates a very stereotypical and simple escape mechanism. The grasshopper rotates and runs forward. Okay? The cockroach. Yeah, grasshopper has different mechanisms. But for this approach to be useful, the cockroach must do it fast and reliably. Now, it doesn't matter to the cockroach whether it's a pink slipper or a brown slipper or a newspaper, or if the attack comes from azimuth of 31 degrees or 31.7 degrees. A simple left or right will do, but this has to be reliably computed and fast. Okay, so in terms of accuracy, what's important is not the resolution, but the ability to correctly discriminate coarse stimuli. And we are also able to identify sound source location, and for sound at low frequency, so when the wavelength is relatively longer than the distance between our ears, we can use the time difference that the sound reaches both of our ears. Okay, so this is termed the interaural time difference, or if it's a specific tone, you can just relate it as the interaural phase difference. And what we'd like to do here in this talk is open this black box and see how does the information about sound source location is encoded in the responses of neurons in the auditory system, and how is it possible to extract information about the direction of sound source fast and relatively reliably. And as you notice in this picture, this is also not a guinea pig yet, but as I told Jonathan before, the only difference for a theoretician between the spherically symmetric monkey and spherically symmetric guinea pig is radius. So, and in certain units it's 1. The reason I put this here is because this is the canonical picture I have in my mind when I talk about a neural code. And I think that you are all familiar with it, and I bring it only to highlight a few points here. So I guess all of you are familiar with it. A monkey is trained to do simple reaching tasks to one out of 8 directions. So he is in front of a computer screen and receives instructions to do simple reaching tasks to one out of chosen directions, 180, 90, and so forth. And while he performs this task, extracellular recordings are being done from his primary motor hand region. And typical results of such recordings are presented here. Okay, so what you see are actually 8 different raster plots, each showing, okay, so the x-axis is time 0 is the onset of movement, and all these 5 tries show the spike of the same cell during 5 tries where he reached the direction of 180 degrees, 135, 90, 45, 0, and so on. Now, if you look, and I'm sure you already have, at the mean firing rate, okay, the mean number of spikes that this cell emits in some time interval around movement onset, you see that it depends on the direction of movement, of arm movement. So it fires with a higher rate when this cell fires with a higher rate when he reaches the direction of 180 degrees, less so when he goes to 135, less for 90, and so on. And the fact, and this is summarized in the bottom curve that shows the tuning curve, so the mean number of spikes, the conditional expectation number of spikes as a function of the direction of arm movement, which is typically characterized with some sort of bell-shaped tuning that peaks at the preferred direction of this specific cell. So the fact that the conditional mean depends on the stimulus implies that there is information about the direction of arm movement that's encoded in the first-order statistics of this cell response. And what limits this information is, of course, the inherent variability, trial-to-trial variability in this of this cell response. So it'll be more difficult to judge whether a single trial originates from 45 degrees than from 0 degrees or minus 45. Another minor point to mention is that usually when we talk about encoding of arm movement, it is traditional to take something about 500 milliseconds to 600 milliseconds and study information that's encoded in this time duration. But nevertheless, we still assume that there is somewhere someone that tells us, okay, start counting now. Okay, so we always have this underlying assumption that it's okay. There will be an onset signal that will tell us when to start moving our hand. But we ignore this simple thing that we never really found it or never really characterized it or how faithful this is. However, it doesn't seem to matter too much. So if we'll jitter a little bit this onset detection, say 10 milliseconds to the right or to the left, it will not change much the result. But the reason it does not change much the result is that they were cheating in the first place. We're taking 600 milliseconds, which in some cases is slightly beyond what is plausible. But what I want to do now is to turn to our guinea pig. So, so this is a guinea pig. And the guinea pigs were anesthetized, and short tone bursts were played to their ears, while extracellular recordings of inferior colliculus cells has been done, and all the data that I will show you in this talk, unless otherwise stated, comes from the labs of Trevor Shackleton and Alan Palmer. And this is how, say, typical results look like. So what you see here are actually 21 different raster plots showing the response of a single cell. So here I present 50 trials where a short tone burst was presented at an IPD of minus 0.2. Okay, so plus is contralateral leading and ipsilateral leading. Okay, and another raster when the IPD was minus 0.8 in T. IPD is the interaural phase difference, so it's in units of cycle here. Okay. Yes. Yes. So okay. So first the best frequency of the cell was characterized, and then the tone burst was at the preferred direction, at the preferred frequency of the cell. So of course, if you take some time window around, say, around the response of the cell, you can clearly see that around 0.16 the spike density is higher, and this is summarizing the tuning curve of the cell. So the first order statistics, yes, the mean fine rate of the cell as a function of the interaural phase difference is, again, as we expected, is tuned as a single peak, prefers contralateral leading, and decays as the IPD is being further away from the preferred IPD of the cell. But you can...
And why it's not completely to pi?
Why it's what?
Why is it from minus 1 to
Okay, so what they did...
This is in phase, in rate.
Yeah, so in cycles. So it's minus 1 to 1. So what they did is, in order to get a lot of measurements, so this is an outstanding data set. You have here 21 different stimuli. For each stimulus, you have 400 repetitions. You cannot do it from minus 1 to 1 with this resolution. So they did first sort of coarse scanning. They identified the range where the preferred IPD is, and then they scanned, they defined. Yes, Ali? Okay.
The fact that the guinea pig head is small and at the relevant frequencies, the guinea pig will never hear much beyond 1/10 of the cycle, or middle cycle. So the relevant part, the part of the curve is relevant to the IPD is a very narrow part near zero.
But if you look at the mean rate, if you had done the measurement, it won't be roughly symmetrical.
It would be. It's not exactly symmetrical. It goes down more slowly, and its width is about 1/3 of the cycle.
That's right. So I'm saying,
yeah,
the cell will respond.
Yeah,
Yes.
it would respond beyond that, but this is the relevant part for the behavior of the guinea pig.
So even though the cell will respond in other part, it's not relevant to the behavior.
Yeah.
Or for other reasons.
Yes, Josie.
Yeah.
So actually here the stimulus was introduced with time 10 milliseconds, but in the rest of this talk it will be time 0, yes. It's a 50 millisecond time, 50 milliseconds tone burst. In this specific figure it starts at 10 milliseconds, but in the rest of the talk it will be at time 0, yes.
So it takes about 40 milliseconds.
So it takes from here to about here. Well, inferior. Well, first of all, if you look at the top, it's much less. So you have about, say, 25 milliseconds. We'll see more data. Okay, it takes so the cell does respond much slower to IPDs that are not its preferred IPD, and this is something that you can see from this graph. So in addition to tuning of the first order statistics of the rate, it seems that the speed of response, or the latency of the response, is also tuned to the IPD. So you can actually extract information from this aspect of the neural response as well. Okay? And this has led us to suggest a very simple readout mechanism that was termed temporal winner-take-all. So it's sort of a winner-take-all competition, but instead of estimating the stimulus according to the cell that fires the most spikes, it estimates the stimulus according to the cell or the population that fires first spike. Okay, so that makes it inherently fast readout. So we can assume that you have, say, two columns, one with preferred IPD of theta and another with preferred IPD of theta plus some delta theta. And there's a stimulus, and a stimulus drives these two populations, and they start responding. So the temporal winner-take-all estimates the external stimulus by the preferred IPD of the population that fired first spike. Okay, so it's some sort of a race to a threshold competition. Okay, the first one that reaches a threshold, decision threshold, is the one that wins. And of course, the accuracy of such readout will depend on the probability density of first spike time given the stimulus. Okay, so the dependence of this distribution on the stimulus IPD is what encodes information. Okay, so in some sense, we assume that we have two populations, they compete, and in some sense, one population fires sooner than the other. And that enables us to estimate the external stimulus. And the question, though, is, okay, how well can you do that? So one thing that's very simple to do is to do analytic calculation, and we can do it. And when you do this, you can, in the framework of some very simplistic model for the neural behavior, you can find that indeed the probability of correct discrimination of this first spike competition depends on the distribution of first spike given the stimulus. Okay, the tuning in this respect of first spike latency. But more so, when the number of cells in each population is large, it only depends on the initial increase of this density. Okay, the initial it depends only on the distribution of first spike times at very, very short times. And at these short times, you can approximate this function, which could be generally very high dimensional. You can approximate it by three parameters, just sort of a scale parameter, an absolute delay that depends on the stimulus IPD, and some sort of a shape parameter. And we can analyze it using some very simplistic modeling, which allows us to disentangle the different parameters, and we find out that if only the scale parameter depends on the stimulus IPD, then it doesn't matter to have more cells in the population. So here I show you the probability of correct discrimination of this first spike competition as a function of the number of cells in the population. In this case, when only the rate of the response depends on the stimulus IPD, and you can see that it will not affect the accuracy of the decision. It will affect the time of the decision, but it will not allow us to have a more accurate or reliable discrimination.
Can you give us an intuition why?
It's if you think so of if I just scale the responses of all cells, okay, it's you can just instead of doing this, you can just rescale time, okay, and then you're back to where you were before.
Tuning of scale parameter is if there are.
Yeah. So think of.
But I thought that usually it's a matter of noise, and here there is no added potential averaging of the noise.
So if you think of,
Scale is the mean. So.
if only the scale is modulated by the stimulus, so we can think of competition between two homogeneous Poisson processes. Okay. Now you can so one has a rate R1 and the other has a rate R0. Now if you scale both processes by n, it's just like rescaling time by 1 over n. So it will affect only the time of the mean time of the decision, but not the accuracy of the decision. So the ratio of
The time of the decision is forced by what in the model?
by the first spike.
Oh, so that's part of the model. That
Yes.
The first spike you make
Yes.
the decision.
Okay, if the shape depends on the
That sounds like potentially point of improvement, right? That if the system, if there is noise, then you should maybe wait for more spikes.
definitely, and we'll see where we really need to do that. We'll skip the shape. The good news is that if the absolute delay is tuned to the if the absolute delay is tuned to the stimulus, then actually the probability of error of the first spike estimator decays to 0 exponentially with the number of cells. Okay, so this is the good news. Of course, generically, you'd expect that in a population of cells, all of these parameters will depend on the stimulus, and hence you'd expect that the probability of error will decay exponentially fast with the population size. So you'll end up with a very fast readout that has a very, very low probability of error. But the bad news is that we have spontaneous firing in the brain. Okay, so it actually brings us back to the point of the scale of the noise. And what is the problem of spontaneous firing? So assume we have okay. So again, using modeling, you can just take the most caricature model. Assume that we have two populations. One that has a preferred IPD of theta, and for stimulus theta responds with some spontaneous firing rate up to time capital T, and then jumps in response to the stimulus to firing rate of R. While the
it looks in the data that the neurons are completely silent before.
Well, best typical cells usually are. Cells in the auditory system seems also to have very low spontaneous rate, and we'll see it hopefully. And definitely in anesthetized guinea pigs. Okay, so I don't have data from awake animals. So it might be that the spontaneous rate is slightly higher. And the other population responds with the spontaneous rate of R0 up to time t plus tau. So tau is the stimulus-dependent delay, and then goes up to responding at the rate of R. So assume that a single cell fires only rarely a spontaneous spike given a stimulus. The problem with spontaneous firing is that if you have many cells, the number of spontaneous spikes that will be generated in this period will increase linearly with the population size. So when population size is such that the mean number of spontaneous spikes here will be of order 1, then will decay to chance value discrimination. And one can solve this model completely, and you can see that the probability of correct discrimination as a function of the number of cells in the population for a specific model in which I bring 4 different curves with different absolute delay periods. Okay, so when the absolute delay period is 0 milliseconds, so when the competition starts here, okay, the probability of correct discrimination will not reach 1, but at least it will not decay. But as the absolute delay time is non-zero, eventually we'll have an exponential decay to chance level of this discrimination, which is based on the single spike. So what I wanted to highlight here is first that to overcome this problem, we can, one, have a very good estimate of when the population starts responding, not when the stimulus was on, but when the population starts responding. So we filter out all the noisy spikes that originated from this region. And another thing that I wanted to point out, which has some educational purpose, is that this is an extremely simplistic model. Okay, it's non-realistic and it's completely ridiculous in the sense that no neuron that respects itself will behave like this. But, and this is the point that, if you remember one thing, except for the joke I told in the beginning, which was not mine even, I'd like you to remember this, is that this is exactly how we approach theoretical neuroscience. Instead of hiding behind a numerous number of parameters which we have no control over, we try to solve analytically the most caricature model that you can obtain complete analytical understanding of that captures the essence of the problem. Okay? And our ability to generalize to large scale, different system, various conditions does not lie in the in how complicated our model is, which occludes our ability to gain clear understanding of the problem, but on the other hand, it lies on the ability to look at the most simplistic model and obtain complete analytical understanding of the issue. So having said that, we still have the problem of spontaneous functions. And as Chaim suggested, we need to overcome this problem. We just need to raise our decision criterion to a level which is above the level of the noise. So the noise here, okay, in this case, here, were the number of spikes that were generated until time capital T. Okay, so in the specific example that I show here, the mean number of spikes was 1. For a population, I think it was 100 cells. Should be somewhere here. Okay, so what I show you here is the probability of correct discrimination of a latency code readout, but which does not decide by the first spike, but on the other hand, by the nth spike. Okay, so since the mean number of noisy spikes was 1, as we increase our criterion from 1, we very fast reach almost saturation at probability of discrimination of 1. Okay. Of course, the informative section here, the informative region is the spikes that happen between time t and time t plus tau. Okay, so in this case it was 25. So if we increase our criterion too high, eventually we will decay again to chance value. So to summarize the theoretical part, what I've shown you that a first spike latency code can provide fast and accurate readout if the spontaneous rate is relatively low. The spontaneous rate limits our ability to accumulate information fast from large populations, but we can overcome this by either or both by starting our competition at the time of the neural response, and by raising our decision criterion above the noise level. And this is true, at least in theory. So how does it look inside the brain? So this is how it looks like. These are results of 4 typical cells, inferior colliculus, anesthetized guinea pig. And what you see in the first row is the tuning curves. So these are typical tuning curves. They prefer the IPD to originate from the contralateral side. They show unimodal tuning curves. Everything is fine. But we can also look at the temporal structure of the neural response in terms of the PSTHs. And you can see the PSTHs of, say, 21 different IPDs. And you can see that the PSTHs for different IPDs are actually somewhat different. So there is information about the IPD that's embedded in the fine temporal structure of the response. The problem with the fine temporal structure of the response is that it is very, very highly dimensional. Okay, even with this outstanding data set of 400 repetitions per trial, we will never be able to faithfully probe the probability of the really fine temporal structure of the response. On the other hand, if you look at the on the first spike distribution, this is something that is much lower dimensional than the PSTH. You can start looking at with 400 repetitions per trial. If you look just at the density, it's kind of noisy. You can see that it depends which IPD it was, but it doesn't clear how that helps us. But if we smooth it, if you just look at the cumulative distribution, so you just integrate it, everything becomes clear, and you can clearly see that, for instance, this cell, there are certain IPDs that it responds very fast to, and several IPDs which it responds rather slow. Okay, and this is summarized here in these color plots that shows you the cumulative distribution of first spike time in the x-axis in milliseconds, as a function of the IPD of the cell. And you can see that if you think of the tuning curves as level curves of this function, okay, so you can see that there is some sort of a unimodal bell-shaped tuning of the first spike latency to the IPD, and it peaks at something that seems as if it's close to the preferred IPD of the cell as defined by rate. And indeed, if you compare them, if you look at the difference between the preferred IPD that's computed using the rate and the preferred IPD of the latency, you see that the difference is very small. There are 2 cells, which we will call them outliers. So this, everyone call it outlier. This we have reasons to ask you to remember these two cells, and we'll come back to them later on. These are cells that did not show very robust tuning of the first spike latency to the IPD. Okay, so how much information can we extract from the latency of the first spike? So what I show you here are neurometric curves for the same 4 cells that I showed you before. So, and those who cannot see colors will excuse me. So I use the RGB. So the red squares and red lines show the neurometric function of a single cell. When using the when using the temporal winner take all with the first spike, with one spike, the purple or magenta is the neurometric curve computed with the rate in the entire neural response. As you can see, for many cells, here, here, and here, although the accuracy of the temporal winner take all is slightly inferior to that of the entire rate code, it is not inferior by much, and you gain time of computation at a relatively small price to the accuracy. Okay, what I show you in the red, green, blue, and black is the accuracy of the anti-WTA using a criterion of first spike, second spike, third spike, and fourth spike. And typically in many cells, you can see that there is improvement when you go from first to second, from second to third, and so on. You can note that in this case, in the case of this cell, it does not happen. Actually, going to the third spike or fourth spike deteriorates the accuracy of the cell. And what is that so? Okay, so this is just a population statistics. So let's just focus on panel A. What I'm showing you here is the J&D of the first spike readout as a function of the J&D of the rate readout. So we've defined the J&D as the IPD difference between the preferred IPD at 0 and the IPD in which it reached 75% accuracy. Okay? So you can compare the accuracy of rate code and the first spike, and you can see that, okay, first of all, lower J&D means more accurate. Okay? So the fact that the J&D of temporal winner take all is above, so the fact that these circles are above the diagonal means that typically the accuracy of the first spike latency is inferior to that of the J&D. Nevertheless, for many cells, the accuracy is very similar or comparable to that of the rate of the entire response. You will note a few cells which did not reach the decision criterion. These are cells for the first spike latency code. So these are cells that did not show very robust tuning of the first spike latency to the IPD. 2 of them we've already encountered. So bear in mind these 4 cells to the end of the talk. We'll meet them at the end. Here I'm just comparing the accuracy of the decision based on the nth spike. So n is the J&D of the second spike is above that of the third and above that of the fourth. Typically, in many cells, second, third, fourth. In a few cells, it happened the reverse. Okay, so the second, so this is the cell that I show you here. Okay, so the J&D of the second spike is actually better than the J&D of the second, which is better than the third. And we can understand that if you look at the distribution of the nth spike time. Okay, so again, these are each column shows you the statistics of one of the 4 cells we've already encountered. The top row shows the first spike distribution. Okay, so the x-axis is the IPD. Sorry, x-axis is time, and the y-axis is the IPDs. And what happens is when you go from first spike to second spike to third spike, is that if you look at the level curves, or if you look at here, you see that it becomes somewhat narrower. Okay, so the tuning is narrower as we go to as we raise our decision criterion in these cells. Okay, the price we pay, of course, is that the second spike always happens after the first one. So we have some trade-off of accuracy versus computation time. On the other hand, the fourth cell has a very low firing rate. So if you go to the third spike, it almost never happens. So eventually, the temporal winner take all will just decide according to some spontaneous spike that will happen many seconds probably later. So we have actually two problems when we raise our decision threshold using a single cell. One problem is that it's no longer a very fast readout. Okay, and the second problem is that we have some cells that have very low firing rate, and we might just not only filter the noise but also the signal. So to overcome this problem, we need to obtain a fast and accurate readout. We need to look at more cells. So we generate some sort of a cell population from the statistics of single cells, and we computed and we studied how well can a single cell, how well can the temporal winner take all, the first spike latency code, can discriminate based on the response of population of homogeneous N cells. So here you have probability of correct discrimination for two different IPDs. It's actually it's this cell. This is data. This is this one. This is data, extrapolated. So I have a.
You have 10 cells.
Okay. I have a single cell, and I generate a cellular population. Okay, so I just sample it as if it was 10 cells, 10 homogeneous cells. Okay, to overcome in order not to complicate things with heterogeneity as well. And what you see here is what you expect from theory. Initially, okay, you can improve a little bit the accuracy of the first spike code, but eventually, once you have too many cells, which makes the spontaneous firing rate prior to the response of the population to the stimulus, eventually it will decay exponentially to chance level. Okay, and we can see it. We just look at the distribution of first spike, okay, as a function of the IPD of the stimulus for different sizes of populations. Okay, so this is the distribution of the first spike. We've seen it before. This is the distribution of the first spike in a population of 2 cells. So what you can clearly see is that if the first spike happened with probability 1 around 60 milliseconds, then with 2 spikes, it will happen much sooner, around less than 20, between 10 and 20 probably milliseconds following stimulus onset. But it also, okay, so this is the preferred IPD of the cell. And I'm comparing the two curves that I showed you here. Okay, so this curve is a curve that it's harder to discriminate. Okay, the probability of discrimination is lower; it's close to threshold, so it corresponds to discriminating between the preferred and this IPD. Okay, and you can see that also the probability of having a spike here is also increased, and when we reach 10 cells already, the competition is very tight. And the second curve, okay, where it's easy to discriminate the two IPDs, is actually when we are very far from threshold. Okay, so it's between the preferred IPD and this IPD. But what you can see is even if the response to the stimulus happens at about, 12 milliseconds following stimulus onset, when we go to a large population, then the response is dominated by the spontaneous firing, which occurs before the stimulus selective response. Okay? So one way to overcome this problem, of course, is to raise our decision criterion. So what I show you here is the neurometric curve of a competition of 10 cells, okay? And these 10 cells will fire an average of 1 spike before the, an average of 0.1 spike, before the stimulus-specific response. So the neurometric curve of the first spike latency readout eventually will saturate to a probability of 90% discrimination. Okay, in 10% of the time, we'll have an error due to a spontaneous spike prior to the stimulus-specific spike. But as we raise the criterion to 2 spikes, then the probability of having 2 spontaneous spikes in this population of 10, pseudo-population of 10 cells is 0.5%, and you can see that it increases very rapidly to almost 1. In 3 spikes, it's as good as it gets. But just to be honest about it, this is the distribution of spontaneous firing rate in the population. So the cell I showed you has a spontaneous firing rate of 0.8 spikes per second. So it's this cell. You can see him here. But most of the cells actually have much lower firing rate. So 50% of the cells will fire at less than 0.1 hertz. Okay? 2 cells, and only 2 cells in this data set out of, I think, 33, fire have characterized with a spontaneous rate which is more than 10 hertz. Okay, so remember them as well. Okay, we'll see them at the end if we get to it. So we can raise our decision threshold and filter out the noisy spikes that happen before the stimulus-specific response. Alternatively, as I told you, we can actually filter the noisy spikes if we just start our competition not at time 0, the stimulus onset time, but if we do it a little bit later, or at 12 milliseconds in which the cell actually starts responding to the stimulus. So if you fine-tune your first spike latency competition to the exact time in which the cell starts responding, then eventually you will not have the decay that's due to spontaneous firing before the stimulus-specific response in both of these cases. Of course, the problem here is that you need to obtain a very, very reliable onset detector. And in general, a latency code needs an onset detector. And to this end, I rely on your memory of these 4 cells that actually did not reach threshold when we studied their J&D in terms of the first spike latency code. So these 4 cells are shown here. Okay, so cells 5 to 8, each in his own column. So they have some to be very, well, it's somewhat modulated by the stimulus, but not by much. But the first spike distribution actually has very, very weak or no tuning to the stimulus. So if you look at these 2 cells, maybe even this one, the tuning of first spike time to the interaural phase difference is absolutely flat. Okay, so this one is slightly sluggish, but here it is flat. So you actually can use the untuned cells, so the cells that do not encode the information that we really need to compute fast, as onset detectors. You can just use the first spike to tell you, okay, here there was a stimulus, start now the competition. And this is a very simple readout, and you can characterize its accuracy. And the accuracy of an onset detector actually has two components to it. The first and each component has 2 components to it as well. So the first part is the detectability. Okay, so you can characterize it with the probability of hit, which is just the probability that given a stimulus, the cell spiked. Okay, so the probability of hit is actually rather high for these cells. And there's also a probability of false alarm. One thing to mention is that when you're trying to detect onset of something, then false alarm is not probability, but rate. Okay, because when there's no stimulus all the time. There's infinitely many no stimulus at every unit time that you look at. So a cell can have 0 false alarms in every second, or even 5 false alarms in every second. Okay, so this is not a probability. And the other two parameters, the other set of parameters is the temporal fidelity of the onset detection, which again is characterized by some sort of a bias. What is the mean time that you actually, that this detector will tell you, okay, now was the stimulus. So here you have the probability, first spike density marginalized over all different IPDs, but it doesn't depend which IPD in these cells. And the other, the last parameter is just the variability of this estimate. So we have a winner cell that has a hit of 1, false alarm rate of 0. It doesn't mean that it has a 0 spontaneous rate, but the data is finite, and in several seconds didn't fire a spontaneous spike. So this is as good as we can estimate here. And then the question, okay, so given this onset detector, how does it affect the accuracy of the first spike code? And it turns out that with such a reliable onset detector, it does not affect much. Okay, so if you compare the J&D of the temporal winetical of the first spike computation based on the estimated onset versus based on the actual onset time at time 0, you see that more or less there's no change. Okay, and this is due to the fact that the spontaneous rate of the cells in the population is rather low. We have two cells I actually ask you to remember with a fire rate which is above 10 hertz, in which if you use the actual onset time, then you incorporate a large number of spontaneous spikes. And if you use the estimated onset, which is at time 12 milliseconds, then you actually filter them out. So the J&D using the estimated onset is actually lower. It's better than the J&D with the actual onset. But we understand why is that. So to summarize and wrap up, we saw that IC cells are tuned, show tuning of their first spike latency to the stimulus direction. And this is something that's already been known. It was published also in a paper by Eli a few years ago. Showing you that rate and latency for the IPD are very close. The tuning is rather similar in most cells. For single cell, typically the accuracy of the latency code, the first cycle latency code, is comparable to that of a conventional rate code that uses the entire response, and we can improve it if we decide according to the nth spike typically. We saw that spontaneous firing rate limits the ability to accumulate information using a first spike code from large populations because, of course, the spontaneous firing rate. We can overcome this using first a more accurate onset detection, and second, to raise our decision criterion to above the noise level. And finally, showing you that cells that show full latency tuning of the first spike latency actually can serve as onset detector, and think this can be used as a general theme. If you need an onset detector for one purpose, you just need to take the cells that are not sensitive to this purpose. And finally, let's thank my collaborators. So the data I show you here was originated in the labs of Alan Palmer and Travis Shackleton. Eli Nelson gave me many data sets of auditory system. Adam Cohn, I collaborated with him on the visual system, monkey, primary cortex, and then Segev works on the ultrafish. Students, Owen, who is no longer with us, is now in NIH. Worked actually on data from Adam Cohn's lab, and Oran, who worked on this data that I showed you here. So, thank you very much.
Questions? I have a question. Oh, frankly. So the conclusion that the temporal lunatic all performance is roughly the same in rate, except for the time of the. But even that, your results show that depending, if you have many neurons, then rate can improve, and the lunatic all has problems. It can actually deteriorate.
Okay, so
So this suggests that there is some advantage of doing the rate coding, although I don't like rate coding. But if you really want to sample large pool.
So first of all, to be actually fair, at the time of the first spike, there's no real difference between a rate code and a latency code. I think that the main discrimination is whether the decision is done at a certain time following stimulus onset, or whether the decision is done when crossing a threshold, which I think might be more natural to a neural system. The issue of using the latency code was to highlight the portion, the effect of the information encoded in the temporal structure of the latency of the response. If you look at the PSTHs somewhere here, see, or if you look at the distribution of first spikes, you see that at the preferred direction, there's a very sharp peak, very, very near stimulus onset. Okay, and usually there's a spike here at the preferred IPD of the cell. So if you have 10 cells and you discriminate according to the fifth spike, most likely you will do your discrimination here at the best IPD of the cell. Okay?
Well, let me talk about the theory. You showed me 4 cells, I don't know. But you yourself showed that theoretically, if there is spontaneous activity, and you increase the number of neurons, the temporal decoding will have problems.
If you take if you scale the number of cells in the population and you do not scale your criterion, your decision criterion, then you'll have problems. But essentially, if you scale the decision criterion roughly linear with the number of cells in the population, then eventually you will overcome. You will filter all the noisy spontaneous firing, and okay, let's forget this. So if you scale, you know what, let's go back to the theoretical slide. Okay, so here I'm not using the fact that there is that actually the PSTS does not look like that, but it peaks here and then it decays. I'm not using this fact. I'm not even using the fact that here it responds much, much stronger than here. But nevertheless, even here, you have only a single cell will generate, you can calculate it, but never mind. Let's say a single cell will generate a spike here with a probability of 0.01. Okay? So 10 cells will generate a spike. Let's say a single cell will generate a spike with a probability of 0.1. Okay? So 10 cells are likely to generate a spike at this time. Okay? But if you scale the criterion linearly with the number of cells here, okay, then you will filter out these spikes. Okay.
So the neuron that, if it is used, the one that the onset detector gives you a small capital T.
No. This is one way to overcome this, but even so, Chaim is worried. Okay, a single cell will generate a spike with probability of say 10%. Okay, we don't have such cells in our system because we have 20 milliseconds and the fire rate is lower than 1 hertz. It's spontaneous, much lower. But nevertheless, let's assume that we have a probability of 10% of having a spontaneous spike here. Okay. Then the mean firing rate of 10 cells will be 1. Okay, so there's a very large probability that actually, if we decide by the first single spike here, then we just have a chance decision. Okay, what you need to do is you need to scale. So you can, for every, essentially, the mean number of spikes here is R0 times capital T times N. Okay, so you need to scale your criterion that's such that it will be larger than this. Okay, and because typically the response to the preferred direction is much, much larger than the baseline, eventually you have some margin in which it's very, very unlikely that you will affect your decision by the spontaneous firing that happens in this purely noisy response, and you will have a very and you will have a stimulus-specific response that occurs around the mean time of the first informative spike on the cell. Okay?
You mean this N?
Yeah, this yes, the small N. Ellie.
There's another issue here that this is the fact that the individual neurons here have discrimination abilities which are almost the same as behavior. So the best of them are as good as behavior. The typical one would be the GND, which is 2 or 3 times larger than behavior, not animal behavior, but human behavior. So you don't, presumably, to do the behavior, you don't need to collect information about for more than over 10 neurons for this specific class of.
Yes, but on the other hand, if you think of how is this actually being done, implemented in the central nervous system, then I wouldn't imagine that this so-called readout or whatever computes the sound source direction actually pulls information from a specific single cell for every IPD. So probably there is pooling of a population, and then you must scale the criterion. It's more so in the visual system where spontaneous firing is much higher, and the absolute delay time is also longer, and so on and so forth. Yes.
So what's the very simple neuronal model which you use?
What's the
very simple neuronal model which you use?
For what? For what aim? Which
To estimate the probability
here?
of firing.
So
And also,
okay.
what do we know about the inputs to these cells? Are they synchronous or anything?
What?
What do we know about the inputs to these cells? Are they synchronous or anything?
Are they what?
Synchronous.
So it comes from the same two earphones. So I guess it's synchronous. But I don't really. Let's start with the neuron. So this is not a dynamical model for the response of the cell. This is just a statistical model that describes the statistics of the response of a caricature toy model. Okay. So this is just a Poisson process. Okay. Now, what exactly did you want to know about the synchronicity? So there is phase locking. There is these are what? No, this is not. So this is, I think, third station would be more accurate. Definitely, this is something that you should have in mind here.
So is it very sharp, the distribution of first passage time? Assuming that the two different populations just get input which are stronger and weaker, depending on the phase difference.
Okay, so what you ask actually is what is the distribution of first spike time?
Yeah.
Okay, so this is the so what you see here is 20 distributions of first spike time for 21 different IPDs. So this is the distribution of first spike time at the preferred IPD of the cell. You can see it here as well. So
this is data.
Yeah, this is data. This is.
So if you would sort of take a sort of integrating neuronal model.
You integrate?
A model for generating those dynamical model of the neuron.
Okay, so you're asking what kind of model will generate this?
Or what kind of input such a neuron had to generate something.
So, so as you said, an integrated fire. So even if the input was only had only tuning of its rate, and you have some threshold mechanism, okay, so in integrated fire, you will get some sort of a delay that depends on the input. Okay, but I don't want to go into speculations because, as you can see, the data is all over. So what causes this here? What causes that here? there are different various mechanisms that can occur here. As I said, this is something that when you're doing modeling, you should also always ask yourself, why? What is it good for? So with sufficiently large number of parameters, eventually we'll be able to fit each and every point here. Okay, so the question is, why do we want to ask this? And I think, if you look at the temporal structure, we don't have enough constraints to actually say something very intelligent about what causes this behavior. But it will be interesting, nevertheless, if we could probe simultaneously at 2 different stations along the auditory processing pathway. But then you need to probe them to find 2 cells that has the same best frequency.
Behavior-wise, where does the animal decide to do something? I know this
Unless it has, yeah.
But how much time does it take for an animal to act based on this data?
Okay.
Is it possible that they even use rate, that they wait for 100 milliseconds or something?
So,
or do they do action in 50 milliseconds?
I don't have data for a specific sound localization task in guinea pigs, so I can't give you numbers here. Eli told you that the J&D of the azimuthal ability to distinguish different sound source locations is similar to that of the neurometric ability of the good cells. But
But that's about the accuracy, not about the time. You're asking about the time.
Yeah, but we don't have I don't have data about.
In guinea pig, there's no information. In cats, the initial movements, ear movements that are related to motion, are the fastest thing that you can measure in motor tasks. They are something like 50 milliseconds after onset. 50, yes. So these are very, very fast responses, and they cannot. They're so fast that they cannot go through cortex. They must operate through a motor loop that goes in the brainstem, presumably it involves this neuron.
So you all had this experience, well, most of you had this experience, well, not in the auditory system, but you drive and then you see something jumps into the road, you brake, and only after you stop you realize it was just a plastic bag. Okay, so you know that there's onset of stimulus, something happened, it happened here, you already have the motor response, and then you can process the rest of the information. So some things you need to compute fast, and I think that sound source location might be very important for that, and some things you can compute later with more elaborate model for the response.
I'll take the last question. So we would all like to have accurate yet fast coding. So one way of getting fast coding is temporal winner takes all, but then the price...
So that's a fast readout, but not...
The fast
okay.
readout. Yeah, but the price that you pay is that it's inaccurate, so you add more spikes and TWTA. Alternatively, you can start with a rate and look at shorter intervals.
Yeah.
Is there a fundamental difference when you take the limit of large n?
If you take it correctly, then there should be no real difference at the limit. So again, at the time of the first spike, the rate code and the latency code is the same. So if you just think of it as a race to threshold, you just integrate the rate till you reach the threshold. Okay?
But there the decision will be restricted by time or onset, not by the arrival of first.
Yeah, but then again, you can when you do the large n limit, you can scale the time inversely with the population size or something like that.
So basically, the way to think about it is a rate code that we all love and understand, just on very short intervals, very short windows.
So the thing, the latency code, if you want to think about it, For a given population size, there's no real difference. The issue is how you scale what you look at in the decision when you consider larger populations. Okay, so that's. But if you take the limits correctly, then you have the same limit.
Thank you very much.
Thank you.